Now we're going to talk about crosses. So there are different crosses that are performed in genetics to try to figure out the associate genotypes with phenotypes, okay? So when we're talking about crossings, we're just talking about matings. So the first we have is this test cross. And the purpose of a test cross is to determine the genotype of a dominant individual, okay, or a dominant phenotype. So why do we need to do this? So you should remember that homozygous dominant or heterozygous will give you a dominant phenotype. And you need to know what is that second allele. Are these individuals homozygous dominant or heterozygous? To accomplish this, you're going to always mate them to a homozygous recessive. Why? Because if that recessive trait shows up, it's telling, okay? So if the individual is homozygous dominant, then you would expect them to have 100% het kids that are 100% dominant phenotype. But if that individual is heterozygous, then you expect them to have 50-50, okay? So if you get any recessive children, then that means that dominant parent had to provide a recessive allele. And this is a test cross. So you have a dominant phenotype in front of you, and you need to figure out, are they homozygous dominant or heterozygous? You're going to perform a test cross by mating them with a homozygous recessive organism. Then we have a reciprocal cross. The purpose of this cross is to determine if something is X-linked. Okay, so determine if our phenotype is X-linked. So what you do here is reverse the, let's see how best to explain this. So you have a female here, and we're going to change these around a little, who's mated with a male, okay? So here's our female, here's our male's genotype. She's red, he's white. And then you get whatever the offspring are. So what you would do is make her white and him red to figure out if the ratios in the offspring change, okay? So you're going to change the phenotype according to the sex of the organism to see what happens in the progeny, and that will tell you if it's X-linked or not. And our third and final cross is our back cross. This is usually used to define a pure breed. Okay, you want to make a pure line. So what you do is you have your parents here. Okay, so we mated our parental generation. We got our F1s. We then take the F1s and mate them with the parents. Okay, so we're going to take the progeny or their offspring and make them with the parents to try to produce this pure line. And we're going to do this until we have generations and generations of our genotype and phenotype of interest, okay? And then this is usually used in genetics when you're trying to look at very limited traits. But this also limits your data because it's not on a quote-unquote real background, because they have been inbred for so many generations that they're not as diverse as a wild phenotype, okay? Any questions on crosses? Write them down, bring them in. Let's talk about it.